Okay, hello everyone. Today I'll be talking about lecture 10. So lecture 10 is about the endomembrane system, and really there's not much to say about it. It's just the synthesis and modification and packaging and transport of proteins and lipids, and that's point 3. According to the self-assessment, there were a lot of questions about this part. So what kind of transport that is used for specific molecules, and you have to think about all of the different receptors or the proteins and whatnot, and what kind of molecule it is and everything. So but let's break it down. Really that's the most important thing from lecture 10, and I can imagine a lot of questions will arise from it on the colloquium. So let's start here. Let's start here. So molecular transport, you can divide it either into either micromolecules, micro or macro molecules, and I will actually go through macromolecules first, even though the teacher took that the last, because it's a bit simpler than micromolecules. So macromolecules is divided into either exocytosis or endocytosis. So exocytosis just means that signaling molecules are released, or endocytosis that signaling molecules, or not only signaling, but molecules are taken into the cytosol. And endocytosis can be divided into pinocytosis and or receptor-mediated endocytosis. I'll just write receptor-mediated. Now, pinocytosis, our professor said that the main function of pinocytosis is to regulate the cell size, or size of plasma of the cell membrane. So that's the main function. That's why it takes up soluble materials from the environment, etc. And then you have the receptor-mediated endocytosis, and all you have to know really is that there is phagocytosis and that there is clathrin-coated endocytosis. Really not much else. And then in exocytosis, it's either regulated or constitutive. So basically non-regulated. What's important to know the difference between them is that the most common one is constitutive exocytosis. The most proteins are transported directly out of the cell through constitutive or non-selective constitutive secretory pathways. But regulated exocytosis happens when there's a specific signal that comes to the cell, and this in some way triggers exocytosis. So for example, insulin. I don't know actually if constitutive happens more often than regulated, but most proteins are released through exocyte or constitutive. So they don't really need a signal. That's the main. This one needs a signal to release, and this one doesn't need a signal to release protein or signal or whatever it is. Now, molecular transport of micromolecules is a little bit more complex, and that's why this picture is just really, really good. Like, you just have to learn this picture and everything in it. So, when you need to break down micromolecule transport, you need to know first, does it have a protein? Does it require a protein for the molecule to be transported? The second thing is this molecule that is going to be transported, is it being transported against its gradient or with its gradient? And the concentration gradient, concentration gradient is basically from high concentration of a molecule to low. So from many, to, and so this is with, and the other way would be against or uphill or whatever they say. But yeah. Third thing is it coupled to ATP hydrolysis? And when we say a coupled to ATP hydrolysis, meaning that there is an ATP that will be broken down and release of energy directly. And then the fourth thing really that only ever happens to co-transport, but driven by movement of co-transported ion down its gradient. And we will talk about that. Okay, and so these ones are really important actually to memorize, that oxygen and carbon dioxide and steroid and hormones, they can be used in passive diffusion. Or that glucose and amino acids, they can be through facilitated diffusion, etc. Because this will make it much easier during the colloq to just know. And you don't have to think about what classifies a small molecule, what classifies a macromolecule. You just know. If it's this, then it's this kind of transport. Okay, so if we go back here. Now macromolecules. Macromolecules. This is the main division that everybody does, which is either passive or active. I'll just move everything here to... Okay. So either passive or active. So does it need energy or does it not need energy? And the main thing here when we say that something is active, active transport means it goes against concentration gradient. So anything that goes against the concentration gradient, it has to be active because there has to be ATP somewhere. Yeah, and you can look again at this. Solute transported against its gradients, plus, so active transport. So you need ATP in some way. The thing about this one, the co-transport doesn't really use ATP directly. It uses ATP indirectly, but it's still considered an active transport. Yes. So, but if we start with passive, the most common one would be simple diffusion. And in simple diffusion, you don't need any protein in order for the molecule to go down the concentration gradient. So it's simple. But passive can also be facilitated diffusion. And here the difference would be either carrier proteins or channel proteins. Carrier proteins, they carry, they need, so in facilitated diffusion you will transport molecules that could be maybe hydrophilic I mean, because in simple diffusion the only molecules that can do that are hydrophobic. So only hydrophobic molecules can do simple diffusion, and that's because they can diffuse over the phospholipid bilayer, but hydrophilic molecules cannot do that. And so hydrophilic and charged small ions can use facilitated diffusion to be able to transport yeah. So carrier proteins, they use a protein called uniporter. This is a protein, and this uniporter carries one molecule, and it carries this one molecule down the concentration gradient. So it follows the concentration gradient, everything is normal. Now, to channel proteins, we have gated and non-gated. And channel proteins can also be called ion channels. Now, be careful with the words because there's another word called ion pump. And this is, this is not, they're not the same. They're completely different. So ion pump is used in active, but we will come to that. So just think about that. Ion channels, channel proteins, the same. Ion pump, completely different. Or is not used in facilitated diffusion at least. But maybe some differences about gated and non-gated. Non-gated are permanently open, like for example aquaporins, and this just it's faster for water to diffuse over non-gated channels than to diffuse over a plasma membrane. And gated channels can be opened by a voltage. They are gated by either. They can be opened up by a ligand or by changes in voltage or mechanically open up. Okay, so that's passive. Everything here I just talked about is passive. I have talked about ions and stuff like that, but it's still passive diffusion because it's with the concentration gradient. So that's important. Okay, now for the second type of transportation is active. Okay, I'll just move everything here like so. Okay, so active can be now for active transport we have primary and secondary, and secondary can also be called co-transport. Okay, so a secondary transport uses a symporter or a antiporter, and that's the names of the proteins. And symporter and antiporter, they just decide if the molecules are going to the same side of the plasma membrane or to different sides of the plasma membrane. Now, primary active transport uses ion pumps. They are also called ATPases. Okay? Yes. And so these ones use ATP hydrolysis, whilst secondary transport uses energy that has been stored in the electrochemical gradient. So basically what this means is that they use some energy that has been released by active transport. They use that energy, or by a primary active transport, in order to transport two molecules at the same time. So one could say maybe use ATP indirectly, but again, look at the table. It's a minus, so they don't use ATP. Yeah, you get the point. Yeah, that was it actually about lecture 10. The rest you just have to memorize. You just have to know what makes up the plasma membrane. You just have to remember what the endomembrane system does and what organelles are involved, and that's really it. Yeah. And I hope this helps, and goodbye.